Hello, everyone, and welcome to today's broadcast. Mechanisms of antimicrobial bioconversion in environmental and host-associated bacteria, presented by Dr. Terence Crofts, a pathology and immunology postdoctoral scholar in the Dantas lab at Washington University in St. Louis. I'm Dr. Susie Valdez of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I want to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during this presentation. Just click on the Ask a Question box located on the far left of your screen. Type your questions into the drop-down box that appear on the screen. Our speaker will respond to your questions via email. If you have any trouble hearing or seeing the presentation, just click on the Ask a Question box and let us know you're having a problem. This presentation is educational and thus offers continuing education credits. So click on that Continuing Education Credits tab located on the top right corner of your presentation window and follow the process for obtaining those credits. Without further ado, let me welcome our presenter. Please join me, Dr. Terence Crofts. I will now turn the presentation over to him. Welcome, sir.
Hi, thank you for that introduction. And thank you to LabRoots and Susie and Tiffany for helping me get all this together. Like you said, I'm going to be presenting work done by the Dantas lab and done by myself in the Dantas lab when I was a postdoctoral scholar there at Washington University in St. Louis. And I changed my title for my talk today a little bit to Discovery and Characterization of Antibiotic Resistance and Catabolism Mechanisms. And so broadly what I'm going to be talking about is various ideas about the ecology of antibiotics. Before I get into that, I want to start by talking about why antibiotics are important. And so that doesn't really need to be said, but I think it's worth going over anyways how transformational antibiotics really have been. And so the example that I'm showing you right now is this graph showing the mortality rates per 100,000 people across the first two-thirds of the 20th century. And what's really notable is that if you look at the trend lines of this mortality, it's decreasing over time, but there's a peculiar steepening of that decline shown in this box kind of centered around 1940. And what's driving that increase or decrease in mortality rate is the introduction of antibiotics to treat tuberculosis, pneumonia, and other bacterial infections. And so this really drives home the point that antibiotics are an integral part of modern medicine. And this is kind of looking at that history of antibiotics in the new graph. What this graph is showing is in blue, the years when first-in-class antibiotics were introduced, and in red, when resistance was first observed in the clinic. And what I want you to take away from this graph are two major points. The first being that most of the antibiotics that we know and love were discovered as first-in-class compounds over 50 years ago in this so-called golden age of antibiotics. And a second major point to get from this graph is that in most cases, just a few years after they were introduced into the clinic, doctors were able to observe the evolution of antibiotic resistance in bacterial infections, shown in red. So this kind of gets across this point that antibiotics have been very important to medicine, but they are steadily losing ground against antibiotic resistance. And so that's kind of why I want to bring up this idea of antibiotic ecology, or the ecology of antibiotics. And this is kind of a fuzzy way of talking about a few different points. The first one is that a lot of antibiotics we use in medicine are actually natural products or derived from natural products. That is, microorganisms and often bacteria in the environment, the soil, for example, are the producers of these. And I'm showing those here as these little spiky red balls. But that's not the end of the story of antibiotics, of course. once they're in the environment, they're there as a way for bacteria to compete against their neighbors. And so oftentimes their neighbors will develop antibiotic resistance strategies to protect themselves from these otherwise antibacterial molecules. And so I'm going to be talking a lot about the second of those points. And then about the last half of my talk is going to be about this third component of this ecology of antibiotics, which is sort of the back end of things. What happens to these antibiotics in the environment? Can they just sit there? Are they degraded? And it turns out there are bacteria that are able to actually consume antibiotics. And so I'm going to be presenting on my research studying the enzymes and mechanisms and pathways that bacteria use to catabolize antibiotics. But first, I'm going to be talking more about resistance. And the reason for that is that resistance is an important part of the story, but it's also a large problem by itself. So in the last 2 years, we've been able to tally the number of deaths resulting from antibiotic-resistant infections. In years before this, antibiotics would work to cure these infections and save lives, but now more people are dying because the bacteria have grown resistant to the antibiotics that are used in medicine. And it's estimated by some that in the next coming decades, the annual global death toll due to resistant infections could rise as high as 10 million people. And it's not just lives lost, it's also about straining the medical system and hurting economies. These antibiotic-resistant infections are much more costly and time-consuming to treat, even if there's a good outcome in the end. And so compounding this rise in resistance is this decline in the last decade, and especially the last few years, in development of new major classes of antibiotics. So, like I said earlier, the golden age of antibiotics, when most of these compounds were first discovered, was 50 years ago. And while resistance has been increasing, the market for new antibiotics has not been very good. And so a lot of major biopharmaceutical companies have been exiting the antibiotic discovery arena. And I think in the past 2 years or so, actually, another 3 big players have shuttered their natural products divisions or antibiotic divisions. And so it's kind of this problem that's increasing. And so it's worth talking about where resistance to antibiotics is coming from. It's not just rising out of thin air, it needs to come from genes that exist somewhere. And one of the major hypotheses for where antibiotic resistance arises, at least originally, is this thing called the producer hypothesis. And so what this hypothesis posits is that antibiotic resistance originates from those bacteria that are the original producers of antibiotics. And that makes sense because you'd expect that if a bacteria is producing an antibacterial compound, it would need to take some steps to protect itself. And so a lot of these producers actually encode resistance genes that protect the host from the antibiotic that's making that pump it out. And as sort of a fail-safe, a lot of them also have antibiotic degrading or antibiotic modifying enzymes to detoxify any compound that they accidentally fail to excrete. And so this makes a lot of sense. A lot of the clinically used antibiotics that have developed resistance or had resistance developed against them originate from soil bacteria. And if you look at sort of the history of these genes, it's been estimated that the genes for synthesis and resistance antibiotics are ancient and have been around for a long time. And so there's some nice experiments that kind of support this. So one of these is a great paper from the Wright lab, where they essentially took advantage of the fact that permafrost, which has been frozen for tens of thousands of years, provides sort of a history lesson of each era where the ice or the permafrost was frozen. And so what they did in this paper was they dug down to a layer of permafrost that corresponded roughly to 30,000 years in the past, extracted DNA, and then sequenced that DNA in order to look for the presence of antibiotic resistance genes. And despite this being tens of thousands of years prior to the industrial use of antibiotics or even their discovery, they were able to find genes that would encode resistance to beta-lactam antibiotics like penicillin, tetracycline antibiotic resistance genes, and resistance genes to glycopeptides such as vancomycin. So even before humans were using these on a large scale, bacteria had already very thoroughly discovered ways to protect themselves. And so there's antibiotic resistance already in the environment, but that's not to say that humans aren't having their own impact as well. So in this third paper that I'm showing, Nat et al. Were able to look at soil samples collected over the past century and then quantify the abundance of different antibiotic resistance genes over time. And so what they saw is an agreement that before antibiotics were introduced, you're able to find these resistance genes in the environment. But what's very notable and somewhat frightening is that over time, as humans began using antibiotics on a more industrial scale, both medically and in agriculture, you see a concomitant rise in the abundance of these different antibiotic resistance genes, really demonstrating that humans have selected for this in the environment. And so there's the question of how much does this matter? And it matters a decent amount because we interact with the environment a lot. So this is just a nice figure showing a lot of different routes by which antibiotic resistance genes or antibiotic resistant bacteria can travel between different environments. So a great example is we use a lot of antibiotics in agriculture for things like livestock. So antibiotics resistance or resistant bacteria can of course travel from that livestock to humans directly through farm handlers or indirectly through the food that we consume. Or it can also go directly to the environment, either from agricultural runoff or from the manure, and in a sense pollute the environment with those antibiotics, those resistance genes, or those resistant bacteria, and those can come back into contact with humans through a variety of means. So then the question is, do we actually observe a lot of transfer across environments? And previous work by the Dantas lab, summarized in this review figure from one of my papers, really supports that concept. And so what's being shown here is a schematic of antibiotic resistance genes and associated genes that were pulled from either a village in El Salvador or a peri-urban slum in Peru. And what's notable about this is that that highlighted pink region is showing that the same TEM1 beta-lactamase is present across a variety of different environments. And when I say the same gene, I mean that it has something close to 100% nucleotide identity across all these environments. And so you can actually track this gene being passed from bacteria to bacteria through human waste and latrines or animal waste into wastewater treatment, into wastewater treatment effluent. And so you can see that these genes are being shared around, which shows just how important it is to consider resistance in the environment and what antibiotics are doing in the environment. And so one sidetrack before I get into some more results is this concept that it's hard to study antibiotic resistance and other functions of the soil microbiome because of this concept called the Great Plate Count anomaly. And so what this idea describes is this idea that if you took a soil sample and tried to grow bacteria from it, you might get something like this image of a petri dish, where you see a lot of different bacterial colonies, and it appears to be relatively diverse. You can see a lot of different colors, different sizes, and morphologies. But if you were to take that same sample of soil and look at it under a microscope, you might instead see something like 10-fold or 100-fold greater diversity, because something like 99% of bacteria from the soil are not able to grow very easily in the laboratory. So if you want to study antibiotic resistance in the soil, it's necessary to use tools that don't rely solely on culturing. And so what the Dantas lab does and what many other labs do in this area is use this method called functional metagenomics, which I'll describe. Basically, we start with a sample of a microbiome that we want to investigate. So for example, a pile of soil, but it could also be a fecal sample for the microbiota, or other samples that have bacteria in them. And what we do is we extract the genomic DNA from all the bacteria in that sample, to get a metagenomic DNA sample. And because the Dantas lab was interested in studying antibiotic resistance, what we do is we take that DNA sample and shear it down to the size of an antibiotic resistance gene or a few genes, so 2,000 to 5,000 base pairs in size, and then clone that in mass into an expression plasmid. We can then take that expression plasmid, that library of plasmids, and transform them into a library of E. coli. And then this way we get this library of E. coli that encodes often somewhere around a gigabase or more of environmental DNA. So it can encompass millions and millions of genes. And what we do to pull out the genes that we're interested in is we take that library of E. coli and we plate it onto agar plates containing some sort of selective pressure. So if we're looking for antibiotic resistance, these might be agar plates that contain penicillin, for example. And penicillin is normally able to kill E. coli. So any colonies that form on those penicillin plates must therefore contain E. coli that is expressing a penicillin resistance gene from the environmental DNA on that plasmid. And what we can do is we can take these colonies, we can amplify the plasmid DNA out using polymerase chain reaction, and then use next generation sequencing to read all those inserts to find out what they contain. And after assembling and annotating those reads, we're able to see what resistance genes are in that environment. And so in this case, if we selected on penicillin, we'd likely see a lot of beta-lactamases. So the students in the Dantas lab before I arrived had done an experiment like this before this Forsberg et al. Paper. What he did was he took soil samples and analyzed it for the resistance genes to beta-lactams, tetracyclines, amphenocols, and aminoglycoside antibiotics. What he found was that the soil contains a multitude and a high diversity of antibiotic resistance genes, especially for beta-lactams and tetracyclines in this case. And some interesting patterns that we observed with this was that for beta-lactams and amphenocols and aminoglycoside antibiotics, a preferred mechanism in the soil of resistance is through antibiotic modification or degradation. And in contrast, resistance to tetracycline was mostly mediated by efflux pumps. So this shows that the soil contains a multitude of antibiotic resistance genes. Does that mean that all these genes are going to end up in hospitals and be a problem for us? Another finding in the paper was that that's probably not going to be the case. This is a little bit of a tricky figure, so I'll take my time to explain it. But it's basically trying to measure the likelihood of a resistance gene spreading from relatively harmless soil bacteria into a pathogenic bacteria. So what's measured here is how many nucleotides you have to essentially walk away from a resistance gene on a piece of DNA before you run into a gene that's associated with horizontal gene transfer. So transposon or the origin of replication for a plasmid. And what you can see when you compare soil bacteria versus pathogen bacteria and their genomes and their resistance genes is that you don't have to walk nearly as far on a pathogen genome from an antibiotic resistance gene before you run into one of these mobilization elements compared to a soil bacteria. And that really implies that these resistance genes in the soil are relatively locked in place and are not as likely to be spread, especially when compared to the likelihood of pathogens in clinical settings spreading their resistance genes. Now that's the case at large, but there's some specific examples that are very different. So that was all functional metagenomics where you kind of avoid culturing. But if instead you look at the culturable component of the soil, you get a slightly different picture. So this is again another paper by Forsberg and also Gautam Dantas, where they essentially took soil bacteria and cultured them in the presence of very high concentrations of antibiotics. So something on the order of 1 mg per mil, which is a very high concentration considering we usually use 10-fold less than that or even lower concentrations when we are selecting for resistance in bacteria. So this is any bacteria that grows are going to be highly resistant. what they found was that most of the bacteria that come out of this selection from the soil are Proteobacteria, and many of them are related to or actually are close relatives of human pathogens. And so going back to the producer hypothesis I mentioned earlier, if you look at the number of antibiotics that those bacteria, those producers of antibiotics, are resistant to, they're often resistant to multiple classes, but on average, something like 7 or 8. And they have this nice distribution here in the lower left corner, where there aren't very many strains that have an extremely high number of genes they're resistant to. Now, that's in contrast to this graph in the lower right corner, in which we're looking at the number of antibiotics that these multi-drug resistant soil Proteobacteria are resistant to. And so most of them are resistant to over 15, and a plurality of them are resistant to nearly 20 different classes of antibiotic. So this is really showing that these are somewhat worrisome bacteria. And not only that, if you look at the mobility of these resistance chains, can they be shared with other bacteria? It looks like these Proteobacteria are a lot more reminiscent of the pathogens than of the soil bacteria in that earlier figure. And by that, I mean, if you look at the resistance genes that come out, as shown in this graph, anything that is shaded gray is another example of a gene that is found across different bacterial strains and different bacterial species that has something like 100% nucleotide identity, which is highly suggestive of horizontal gene transfer. And so you can see that a lot of these multi-drug resistant, highly resistant Proteobacteria have multiple different antibiotic resistance genes, shown in red, as well as having multiple mobility elements, shown in green nearby. And these are found across a variety of different pathogens, as well as these non-pathogenic soil bacteria. So this is really suggesting that these are bacteria that are worth keeping an eye on. And so I'm going to use that to actually switch into the second half of my talk, because it happens that these bacteria have another really interesting phenotypes, and that is some of these highly antibiotic resistant Proteobacteria are not only able to avoid being killed by antibiotics, they're also actually able to use those antibiotics as a sole carbon source. So that's the second half of this talk, where these are the antibiotic-eating bacteria. So this is another representation of the bacteria that Gautam Dantas pulled out of the soil that are able to catabolize antibiotics. And so you can see that this F1, F2, F3, P1, these are all different strains of soil bacteria. And here in this graph, blue indicates that they're able to grow using that given antibiotic as a sole carbon source, and red indicates that they can't. But what really is striking about this is that most of these antibiotics are catalyzable by most of these bacteria. And most of these bacteria can grow on most of these antibiotics. And in particular, I want to point out for now, right in the middle is penicillin G, which is the penicillin that's associated with Fleming, one of the first major antibiotics discovered. It's actually able to support the growth of all of these strains. And so in the project I'm going to talk about in a minute, the things that I want to answer were, what are the enzymes and pathways and mechanisms that some of these soil bacteria use to pull off this somewhat astonishing feat of eating antibiotics? And then also I want to answer the question of, are these bacteria sort of professional antibiotic eaters? Or is this more of a neat trick that the bacteria are able to learn and essentially evolve into? So for this study, I focused on 4 Proteobacteria from that soil selection termed ABC for antibiotic catabolizing bacteria, ABCO2, ABCO7, ABCO8, and ABC10. And then for the antibiotic, I focused on penicillin, which is able to support the growth of all 4 of those bacteria. And some other interesting things about penicillin are that it's a natural product antibiotic. So it's actually produced by soil microorganisms. So the soil bacteria likely have evolved alongside penicillin for eons. We know a lot about how bacteria resist penicillin. It's mostly through the use of beta-lactamase genes, which are enzymes that catalytically break apart the beta-lactam warhead of penicillin. So here in this picture where the red atoms meet the black atoms, that's the beta-lactam ring. And the amide bond there is actually where beta-lactams hydrolyze that ring, rendering it non-toxic. And then finally, it's worth noting that, again, penicillin is one of the first antibiotics that was used widely in agriculture and medicine. And so not only have cyanobacteria likely been exposed to it for millennia, over the past 100 years or so, they've been exposed to especially high concentrations of it. So it seems like a very good candidate for exploring antibiotic metabolism. And so to talk a bit about these strains, I don't want people to worry too much about the phylogeny of these strains. They're Proteobacteria, and they are relatively close relatives of known human pathogens, but I'm not going to make a big deal about them, and mostly we'll be referring to them by their strain name. But just so everyone can be oriented, here's the phylogenetic tree, and ABC07, which I'll be talking about a lot, is actually a Pseudomonas. So across the bottom here, I have 4 graphs where we were simply trying to recapitulate and verify that initial finding that these bacteria can grow with penicillin as a sole carbon source. So these 4 strains were grown in mineral media, where the only carbon source available to them is penicillin, where the increasingly blue colors correspond to increasingly high concentrations, shown here maxing out at 1 gram per liter carbon source. And what I think is really fun about these graphs is that they kind of are backwards of what we usually expect, where we see increasing growth the more antibiotic we had. What's not shown is this actually continues the pattern up to 4 grams per liter or up to even 8 grams per liter in some cases penicillin. So not only is the penicillin not killing the bacteria, it's in a sense making them even stronger. So starting off on this project, the hypothesis that I started with is basically as follows. I'm showing you here a kind of cartoony schematic of an antibiotic. And what I hypothesize is going to happen during antibiotic catabolism is that the first step is going to be detoxification of this antibiotic, sort of a safety first approach, because if the bacteria is going to be killed by it, it's not going to be around to be able to eat the antibiotic later. So that's what I thought would happen first. And I hypothesized that the second step of antibiotic catabolism would be this metabolic hydrolytic activity, where a carbon source can be cleaved off from the antibiotic. And because a lot of these antibiotics are natural products, I hypothesized that this catabolic hydrolase enzyme would probably act in a sort of retro biosynthetic way. That is, penicillin, for example, is made up of phenylacetic acid and a pair of amino acids shown here in blue, red, and black. And so penicillin catabolism, hypothesized, would proceed through breaking apart that into the 2 amino acids and phenylacetic acid. So the next step is to find out what are the enzymes involved. And so one of the first things that we did was we performed whole genome sequencing on these 4 antibiotic catabolizing strains to find out what sort of functional limits we can place on what genes they encode. And one of the things that really stuck out at us when we looked at their genomes was that all 4 bacteria encode a somewhat high number of beta-lactamase genes, either 8, 9, or 10 beta-lactamases, with ABC07 having the most. And so this strongly suggests that, in these strains at least, resistance to penicillin occurs through action of a beta-lactamase enzyme. Again, where that beta-lactam ring is hydrolyzed at the bottom of the screen to produce this non-antibacterial compound called benzyl penicilloic acid, and that'll be a bit important later. So that suggests the first step, but what about the next steps? The way we went about finding what the next steps of catabolism might be was by using transcriptomics and RNA sequencing specifically. So what we did was we took each ABC strain and grew it in minimal media, where the only carbon source present would be either penicillin or a simpler metabolite like glucose or histidine, where we know how that metabolite enters central metabolism. And what we did was that we simply compared the ratio of the gene counts, the gene expression during growth on penicillin compared to glucose, for example, to find out what genes are highly expressed, and then find out which of those are the most statistically significant. And so that's the result I'm showing you here. And it's hard to read these numbers, but they represent the fold upregulation during growth on penicillin compared to growth on glucose or histidine. And what was really striking when we looked at these highly expressed and significantly differentially expressed genes was that they were similar across all 4 antibiotic catabolizing bacteria. So all of them have expression of beta-lactamases in blue, and they all have expression of a whole suite of genes in green that together encode this operon called the phenylacetic acid catabolon, which is responsible for allowing bacteria to use phenylacetic acid as their sole carbon source, which again is somewhat of a telling result. And then what was also really interesting was that in each of these strains, there are also highly expressed, significantly expressed genes that were predicted to be annotated as hydrolases or amidases or aminohydrolases, and these are shown here in red. And at least in 3 of these strains, you can see that the amidases and hydrolases are syntenic with the beta-lactamase. That is, they're right next to that beta-lactamase gene, which is very interesting and suggests that they might be important for the metabolism of beta-lactams, like penicillin. So given this, we decided to focus on ABC07 and try to narrow down and, again, verify that these genes are important. So we did a few more RNA-seq experiments. So what we first did was we again looked at the relative upregulation of this suite of genes shown here across the bottom when ABC07 was grown in the presence of penicillin compared to glucose. So just like that first experiment, but we repeated it and again got the same result where these genes are 10-fold to 100-fold to nearly 1,000-fold upregulated during growth on penicillin compared to glucose. And down on the right hand, bottom right corner, is the housekeeping gene RPOB, which you can see is expressed at similar levels across both conditions as a control. And what's interesting is when we instead look at upregulation in response to growth on benzyl penicillic acid, we see some interesting results. So the first thing is in the lower left corner, that first beta-lactamase goes from being highly expressed at 100-fold greater levels in penicillin compared to glucose, down to equal expression in benzyl penicillic acid compared to glucose, which kind of makes sense if that's the main beta-lactamase that is being used to resist penicillin. Because we're feeding benzyl penicillic acid now, that's not an antibiotic. It doesn't even have a beta-lactam ring, so there's no reason to express a beta-lactamase anymore. On the other hand, this middle contig consisting of a beta-lactamase and 3 of these red genes, aminohydrolase, import pump, and amidase, those are still highly expressed in the presence of benzyl penicillic acid. And of course, the entire phenylacetic acid catabolon also remains equally highly expressed with benzyl penicillic acid compared to glucose, as it did with penicillin compared to glucose. What's interesting again is that when we instead looked at growth on phenylacetic acid compared to glucose, now that those beta-lactamases are both downregulated, but so are the amidase, MFS pump, and amidase called PUT2, MFS, and PUT1 here in the lower left corner. Those are all now significantly downregulated, but the phenylacetic acid catabolon is still fully expressed. So this kind of again suggests that there's this stepwise metabolism going on where you have a beta-lactamase leading to these amidases, which potentially are hydrolyzing the penicillin into metabolites. including phenylacetic acid, which in the phenylacetic acid catabolon routes to the central metabolism. And so the question at this point that I was asking was, what are these PUT1, PUT2, and MFS enzymes doing? And so I called these the PUT operon for penicillin utilization. So the first step to answering what are these enzymes, what are these genes responsible for, and verifying their importance was to make some knockouts in ABCO7. So we made two different ABCO7 strain knockouts. The first one, which will be represented by blue line, we knocked out the entire PUT operon. And then the second knockout, we knocked out this gene called PAAF in the phenylacetic acid catabolon. This gene encodes an enzyme that is actually the first to act in the phenylacetic acid catabolic pathway. So without that enzyme, the bacteria should no longer be able to grow on phenylacetic acid. So the first experiment we did was verify these strains still grew well under normal conditions, so with glucose as a carbon source. And what we see in this graph is that, yeah, these knockouts aren't affecting the strain's metabolism generally. They're still able to grow just as well as wild type. And then as expected, the PAAF knockout, the phenylacetic acid knockout, can no longer grow when phenylacetic acid is a sole carbon source, as shown by the red line here. There's no growth. Interestingly, and maybe not unexpectedly, the PUT operon knockout is able to grow just as well as wild type on phenylacetic acid. So there's no downstream effects, and it's not dependent on that pathway, so that's useful to know. So the real interesting results came when we tried to grow these strains on penicillin or benzoyl penicillic acid. And while wild type ABCO7 is able to grow using either of those carbon sources, in this case, neither knockout was able to grow under these conditions, which really demonstrates that the PUT operon and the phenylacetic acid catabolon are both necessary for the growth of ABCO7 on penicillin and penicillin-like compounds. So the next step that I took was to try to nail down what exactly are these PUT operon genes responsible for. And so I focused for now on the PUT1 gene. And the way I went about studying that was I expressed it heterologously in E. coli and purified the enzyme for study in vitro. And what I'm showing you here is what I predicted the reaction would be, that it takes penicillin or benzoyl penicillic acid and liberates the phenylacetic acid from that, allowing that phenylacetic acid to go to central metabolism via the phenylacetic acid catabolon. And so I show you this first graph is my attempts to measure the mechanism and kinetics of this reaction. And so the blue lines represent my positive controls. And what I'm measuring is hydrolysis of an amide bond here. And so you can see that the dotted line, blue control, which corresponds to a beta-lactamase enzyme, it shows really good mechanism and kinetics. And that's not unexpected, because we know beta-lactamases are great at cleaving the amide bond and the beta-lactam ring of penicillin. Now, my other positive control is this enzyme called penicillin amidase. And this enzyme is interesting because it's actually used industrially to remove phenylacetic acid from penicillin in order to make semi-synthetic analogs of penicillin. You can actually remove that phenylacetic acid and put on modified side chains to get things like ampicillin or amoxicillin. And so it's a perfect mimic of the activity I'm looking for. And you can see again that it has very good kinetics with penicillin as a substrate. On the other hand, PUT1, which is shown in that red line, looks essentially identical to the no enzyme control in this experiment, which really is indicating that PUT1 does not have much activity, if any, with penicillin as a substrate. And so this is a little confusing because we thought that we had a really good hypothesis going into this experiment that really supported this idea that PUT1 would act to remove phenylacetic acid from penicillin. But when we thought about it some more, it actually makes a lot more sense that it should remove the phenylacetic acid group from benzoyl penicillic acid. And that's because penicillin, which is toxic to the cells, is most likely almost immediately detoxified by beta-lactamases. And so PUT1 might not even see any penicillin, might only see benzyl penicillic acid as a substrate. And that's what I'm showing you in this second set of kinetics experiments. So here, there's no beta-lactam ring anymore because we're using benzyl penicillic acid. And so the beta-lactamase control is no longer positive control. It shows no activity. The penicillin amidase in the solid blue line still shows some decent activity, but what's really telling is in red is that the PUT1 enzyme is now showing good activity using benzyl penicillic acid as a substrate. So it really does appear to be able to break that amide bond. And just to confirm this, I use quantitative liquid chromatography coupled to mass spectrometry to measure the degradation of benzyl penicillic acid in reactions containing PUT1 or penicillin amidase or no enzyme as control. And you can see, especially at the long time points, that in red, PUT1 is able to remove essentially all or degrade all the benzyl penicillic acid in the sample to a much greater extent than penicillin amidase. So this really indicates that PUT1 is having this activity of releasing phenylacetic acid from benzyl penicillic acid. So I started out with this hypothesis that catabolism would flow through resistance and then retro biosynthetic hydrolase activity. And that's really supported by what we found out here by looking at the sequencing data and growth data and in vitro data, that penicillin is detoxified first by beta-lactamase to make benzyl penicillic acid. And then PUT1 and possibly PUT2, which I didn't test, is able to use that benzyl penicillic acid as a substrate and remove phenylacetic acid from it. Then phenylacetic acid is sent to the central metabolism via the phenylacetic acid catabolon. So I think we have a lot really good evidence for that. But the last experiment that I wanted to do to really nail home the viability of this strategy for bacteria was to say, now we have this pathway now that we think is sufficient to allow bacteria, these ABC strains, to eat penicillin. Can we put that in E. coli, for example, as a proof of principle and make that E. coli into an antibiotic metabolizer as well? So one thing that we need is the phenylacetic acid catabolon. There's actually a more obscure strain of E. coli called E. coli W, which has a fully functional phenylacetic acid catabolon. So what I did was I took that strain and I transformed it with a vector containing a beta-lactamase, which is another important component of antibiotic catabolism in the ABC strains. And I also had that vector express the gene for the penicillin amidase, that industrial enzyme that I showed you earlier that's really good at removing phenylacetic acid from penicillin. So this strain in theory has all three components that are needed to be an antibiotic or a penicillin eater. And indeed, that's what I saw. When I grew it in minimal media with penicillin as a sole carbon source available, I see the same sort of pattern where as you increase the concentration of penicillin, going to darker and darker shades of blue, I saw better and better growth of this E. coli strain. Now, in contrast, in grayscale are the data points for the E. coli strain that has all three components, has two of the components. It has a beta-lactamase and the phenylacetic acid catabolon, but it does not have an amidase. And so that strain is not able to grow with penicillin. Similarly, if I express the PUT operon instead of penicillin amidase, I again see that that is sufficient along with the phenylacetic acid catabolon to allow E. coli to grow better when using benzyl penicillic acid as its carbon source. So to summarize kind of what we learned in these experiments, it seems that this is the approach ABC07 uses to eat penicillin. We hypothesized that penicillin enters the cell normally through pores like it does for many other types of bacteria. And once it's in the periplasm, it's met by a secreted beta-lactamase, which converts it, detoxifies it to benzyl penicillic acid. Now, we actually aren't sure how benzyl penicillic acid gets to the cytoplasm, which is where it needs to be because PUT1 and PUT2 aren't predicted to have a secretion signal, so they're likely cytosolic. Now, I didn't get into this very much, but the Proteobacteria does include this gene annotated as an MFS import pump. And so one hypothesis that we have is that this might function to allow benzyl penicillic acid to cross the inner membrane to the cytosol, where it can be used as a substrate by PUT1 to produce phenylacetic acid, which can go into central metabolism. So I want to contrast this a little bit with the engineered E. coli strain that I produced. And in this case, penicillin amidase is actually also secreted to the periplasm. So when penicillin is in the periplasm, it can be directly converted to phenylacetic acid plus the beta-lactam ring by penicillin amidase, or it can be detoxified by a beta-lactamase to benzyl penicillic acid, following which the phenylacetic acid can also be removed by penicillin amidase. And the way that gets into the cytosol, where it can be used in central metabolism, is that the phenylacetic acid catabolon actually does encode a phenylacetic acid intermembrane importer. So to kind of wrap things up, what I want people to take away from this talk is that, we know that antibiotic resistance and antibiotic resistance genes are rife in the soil environment. However, most of these soil bacteria contain resistance genes that are not strong candidates for horizontal gene transfer or mobilization. The notable exception, of course, are these highly multi-drug resistant Proteobacteria, which do in fact have highly mobilizable resistance genes. And these bacteria are particularly interesting because many of them are not only resistant to antibiotics, they can also catabolize them. And I hypothesize that catabolism of these antibiotics proceeds through detoxification, followed by hydrolysis. And we show that in APC07, this is the case. And so that kind of answers that first major question I wanted to ask in this project, which is, what are the pathway enzymes used? The other question I asked was, are these enzymes sort of professional antibiotic metabolism enzymes, or are they enzymes with other functions that are moonlighting in this capacity? And the results that we found suggest that it's a mixture of that. You, of course, have a beta-lactamase as the first step, and that is a professional antibiotic resistance enzyme. And then the phenylacetic acid pathway is also important for that. And those are, again, professionally used in that capacity by the bacteria. Now, linking those two are these PUT1, PUT2 enzymes, which seem like they are probably enzymes that have promiscuous activity and are more moonlighting in this role. And so one of the obvious future directions that I'm going to take this is asking, does this general strategy of detoxification followed by hydrolysis work for other antibiotics that bacteria can eat? And so an example of that might be the aminoglycosides, which are largely composed of sugar-like subunits. And so with that, I'd like to make sure that I want to thank everyone in the Dantas lab who did this research earlier and where I did my own research, as well as alumni from the lab and our collaborators. And then also acknowledge my funding sources for my time as a postdoc in the Dantas lab at the NIH. And with that, I'm happy to take any questions.
Thank you, Dr. Crofts, for that informative presentation. We will now start the Q&A portion of the webinar, and we'll address some of the most commonly asked questions by our viewers. If you have any questions you'd like to ask, please do so now. Just click on that Ask a Question drop-down box located on the far left of your screen. Type the question into the box and click the send button. Our speaker will follow up with your questions via email. So let's take a look at our viewers' questions. Dr. Crofts, do the ABC strains actually catabolize antibiotics in the environment?
So, right, this is the question that I've gotten a few other times. And it's a sort of a hand-wavy answer, unfortunately. So, as you might imagine, there aren't piles of penicillin sitting in the soil for bacteria to eat. So there's not going to be bacteria in the environment that are subsisting solely on penicillin, although there are some rivers that are contaminated by pharmaceutical runoff that have been found to have very high levels of antibiotics and other antimicrobials. That being said, a lot of bacteria are able to eat glucose, even though there aren't large blocks of glucose in the soil. So I think what's likely happening is these bacteria are subsisting on other components of the soil that are likely aromatic fatty acids, so things that look like phenylacetic acid. And penicillin and benzoyl penicillic acid both kind of fit into that molecular class. So probably what's happening is the bacteria are subsisting on these other compounds, but when they run into penicillin in the soil, they're able to take advantage of that as part of their carbon source. And then when we grow them under laboratory conditions and force them to use penicillin as their sole carbon source, they are up to that task because they have these enzymes which have this sort of cross activity.
Thank you for that. Can we use these phenotypes to fight antibiotic resistance?
So I think one of the cool things about these results is this idea that we can engineer bacteria, including E. coli, to catabolize antibiotics. Like I said a minute ago, there are certain places where wastewater from pharmaceutical manufacturing plants are heavily contaminated with antimicrobials, including a variety of antibiotics. And at the moment, because those wastewaters aren't being processed or cleaned up, those antimicrobials are going out into the environment where they can select for resistance in bacteria that they come in contact with. So one neat aspect of this research is that, we now have this proof of principle that we can make E. coli into an antibiotic-eating strain. There might be a potential use for that where it can be used to bioremediate antibiotics that are spilled into the environment and kind of lower their levels. Now, of course, you don't want to be adding more antibiotic resistance genes in these antibiotic eaters into the environment. But as a proof of concept in terms of engineering, this could be a potential strategy for cleaning up those wastewaters, for example.
Thank you. And Dr. Crofts, is catabolism always connected to resistance?
Right. So this is kind of gets at the idea that in ABC07 and a few other strains we saw, we observed that the PUT operon includes both these amidases and aminohydrolases, as well as a beta-lactamase. And so in the case of ABC07, those genes are physically linked. They're next to each other. But then also PUT1 can't use penicillin as a substrate. It needs the beta-lactamase to act upstream of it to convert penicillin into benzoyl penicillic acid. So it's functionally connected also. So in that case, resistance and catabolism are intertwined and can't really be separated. Now, that might not always be the case per se. So if a bacteria has an intrinsic resistance to an antibiotic by lacking the target of the antibiotic, for example, then you might be able to get catabolism without needing to have an antibiotic resistance gene in place.
Thank you for that. We have one more question. What happens to the other parts of the penicillin molecule?
Right. So in these experiments, we zoomed in on the phenylacetic acid group of penicillin as being the important part in the metabolism, but that's just about half the molecule. So you have the phenylacetic acid group, and you also have the beta-lactam group that's cleaved by the beta-lactamase and then essentially just thrown away. So we have looked at ABC07 and the other antibiotic metabolizing strains to see if they're able to use the beta-lactam ring as a carbon source once you remove the phenylacetic acid group from penicillin. And as far as we can tell, none of the bacteria are able to grow using that. So it seems like the bacteria are just removing the phenylacetic acid to eat and then throwing away the rest of the molecule.
Thank you. And Dr. Crofts, thank you so much for today's presentation and for your important research. I'd also like to take the time to thank LabRoots for making today's educational webcast possible. Before we go, I want to remind everyone that today's webcast is available for on-demand viewing until December 2018. You'll receive an email from LabRoots letting you know when the webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's event. That's all for now, and thanks for joining us. We hope to see you again soon. Bye-bye.
Thank you.